Welcome back, friends. Welcome back in another video tutorial from microbiology with Shagnik. So in that video tutorial we are going to discuss about directional cloning. So I think you know what is the basic concept of cloning. If you know what is the basic concept of cloning, then you can continue it. Otherwise, I will recommend you to go into my YouTube channel and find out the basic big picture of cloning. That will help you to understand what is the basis of cloning, and it will in turn help you to understand the directional cloning. Okay, although I am going to discuss about the basic cloning a little bit short way, so that you can take directional cloning easily. Okay, so what is the big picture of cloning? So cloning means xeroxing, right? We are xeroxing our gene of interest, right? And what is the source of that gene of interest? The source of that gene of interest is natural source, right? From that natural source, we cut out our gene of interest from by restriction endonuclease. Then that gene of interest we insert inside a vector, and then we transform them in a host, right? That is the basic idea of cloning. And for inserting gene of interest in a vector, we need to digest the vector with the same restriction endonuclease that we have applied to isolate our gene of interest from the natural source, right? That is the concept of normal cloning. So in that normal cloning there is a problem. What is the problem? That is that I think you know about ORF, that is the open reading frame. So what do you mean by open reading frame? Open reading frame is nothing but a span of DNA sequence between the start codon and the stop codon, right? That means it is the portion that will be transcribed. Okay, so just think about that. This is the natural source, and this red marked portion is our gene of interest. Okay, for that gene of interest, the direction of ORF is this 5 prime to 3 prime. That means what? RNA polymerase will incorporate here and start transcribing in that way, right? That ultimately leads to the production of RNA, then translation, and then we have our protein. Okay, so if it is, then there is a chance, remember, in normal cloning that our ORF can be altered. How? Our gene of interest will be digested by the restriction endonuclease, which will generate a staggered end, like that, right? Now this staggered end can be attached with the vector in two possible combination, right? Either it can be attached from that position, right, or either it can be attached from that position, right? Both things can be possible. That means our ORF, the direction of the ORF can be changes, right? It can be from 5 prime to 3 prime, it can be changes itself from 3 prime to 5 prime, right? And as here we are relying on vector for our transcription and ultimately expression, RNA polymerase will consider the 3 prime to 5 prime end of the ORF as 5 prime to 3 prime. As a result, what will happen? As a result, our entire ORF sequence will be changed, and as a result we cannot get our protein of interest. Okay, so that is the drawback of normal cloning procedure. So now, how to get rid of those problems? So the solution of that problem is the directional cloning. Okay, as its name suggests, here our goal will be to keep the direction of ORF intact, right? That means the direction of what it is in case of the natural source will be maintained in case of our vector 2. So that is the need of directional cloning, and what is the job of directional cloning I have mentioned, right? It will insert your gene of interest without altering the direction of ORF. Okay, now let's see how it can be done. So before going to talk about directional cloning, I want to talk with you about two important things that you need to know. That staggered cut, I hope you know about staggered end. Okay, let's talk about is in a little bit details. That restriction endonuclease can generate two type of cut. One is blunt cut, another one is staggered cut. Okay, and now that staggered cut can be out of two types. Okay, one kind of staggered cut can generate 5' staggered end, and another kind of staggered cut can generate 3' staggered end. Okay, how? Let's see this. So here we have used PST1 enzyme that generates 3' overhang, as you can see, right? And in EcoR1 we had 5' overhang. Okay, so that is the important thing that you need to know for understanding the basic concept of directional cloning. Now let's move to the topic. Okay, so in normal cloning we are using only one restriction endonuclease that we have used to cut our gene of interest from the natural source along with our vector, right? So here we will use for directional cloning one 5' end, 5' staggered in generator, and one 3' staggered in generator. Okay, that is the idea. In directional cloning we use two restriction endonuclease. Among them one will be 5' staggered in generator, another one will be 3' staggered in generator, and make it very clear that what kind of restriction endonuclease you will use. Your vector along with your gene of interest should contain their restriction site. Okay, that means just using EcoR1 and PST1 will not going to solve your problem. You also have a particular restriction site for EcoR1 in both your gene of interest and vector, along with the cut site for PST1. Okay, then you can done directional cloning. So here I have used EcoR1 PST1. That does not mean that EcoR1 and PST1 is fixed. That is on you, that what kind of gene of interest you are using and what kind of vector you are using and what is the sequence of your gene of interest, right? So again here I want to mention an important term, that directional cloning of unknown sequence is little bit difficult, right? Here we have to take the help of linker, adapters, etc. Okay, that means without doing any kind of modification you cannot perform directional cloning. That is another important thing that you need to keep in your mind. Now suppose this is your gene of interest, okay, and in the two end of your gene of interest you have a cut site for EcoR1 and a cut site for PST1. Okay, there can be another cut site too. It is depending on you what kind of restriction endonuclease you have to choose. Okay, so now we have added EcoR1 PST1 and it will generate a T-shaped product. Okay, similarly we have digested our vector 2, okay, in that way. So After digestion, the vector will generate something like this. Okay, now just by looking A and B, just think about, can now there is any chance of altering the ORF or altering the directionality of ORF? No, because this is the only stable conformation that can attached, or I should say inserted into that kind of vector, right? Because there is a gap for only a T-shaped compound, right? And the complementary bases that we have will only allow this conformation, as you can see. So as a result, if we use a 5' incurt and 3' incurt restriction endonuclease for our cloning purposes, then it can solve our problem regarding ORF. Okay, it will maintain the direction of ORF as it is in case of the natural source, right? So that is the basic concept of directional cloning. By using this kind of cloning, the gene of interest directionality will be maintained here accurately. So that is the basic concept of directional cloning. Idea is exactly same, but here instead of using one enzyme, we are using two enzymes. One is 5' staggered cut, another one is 3' staggered cut. Okay, and the important thing that I want to mention again, that the 5' and recognition site and and the recognition site for the 3' staggered cut should be present in both our gene of interest along with our vector. Okay, otherwise we cannot choose that two combination of enzyme as our restriction endonuclease for directional cloning. Okay, so that's how direction cloning can help us to maintain our directionality of ORF. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends and subscribe to my channel to get more videos like that. So thank you for listening to this class.